Hi everybody, thank you for coming to this symposia. Today I'm going to talk about the possible effect of organ and tissue size on cell survival after cryopreservation by freezing or vitrification. I'd like to start by introducing myself. My preferred place to do this is at the conference hotel bar, or if that's not possible I can be flexible and go to another bar. However, as we cannot meet in person this year, I thought it'd be useful to give some background to myself briefly, so you have some idea at least of whose voice this is. This is me in a posed picture in my lab. My science background started with my undergraduate study in physics. However, I only really got involved in the cryobiology world in 2011 at the start of my PhD at University College London. During my PhD, I worked on large volume cryopreservation of a bioartificial liver device. And following my PhD, I went to work for Asymptote, who at the time were a small company offering consultancy and producing equipment for controlled rate cooling. Asymptote was acquired by General Electric Healthcare a few years ago, before being spun off as part of a larger group now called Cytiva. Day to day, I have a focus on regenerative medicine cryopreservation, whether that be for T-cell therapies and other immunotherapies, and for more broad cryopreservation of tissues and also more complicated organoids in a range of cell systems. Now, that's enough about me. Let's get onto the talk proper. I'm going to start by giving a brief history of cryopreservation when applied to larger structures. There have been a few talks in this session about vitrification, so I'll put a specific emphasis here on some of the slow cooling research where ice forms in the system, although I will mention both techniques. From there, I'll look at the state of the art with organoids and cell therapies, which are a current problem and where a lot of active research is taking place, and that will bring us onto the current research with organs and larger intact tissue. Before wrapping up, I'll then look at what is the limit with larger volumes and where the maximum size is for relevant techniques. I'd like to start with covering what I mean by larger sizes in this talk. When I refer to the size of tissues and organs, what I'm referring to is how big a continuous piece of a biological is. For example, the smallest would be a cell suspension, and from there systems of cell spheroids, moving to larger organoids, and finally tissues and whole organs. What matters is the size of the biological unit. Larger size doesn't necessarily mean, in this context, a larger total volume. When scaling up freezing of cell suspensions from say a 1 ml cell suspension in a cryovial to a 50 ml cell suspension in a cryobag is worthy of a presentation in its own right. For the current discussion, when I say larger unit, what I mean is a larger continuous biological material. The challenges here are of a completely different kind compared to those based when scaling up a single cell suspension. When it comes to freezing larger structures, we already have examples all around us in the natural world. The most numerous of these are plants and trees in temperate and colder regions. Of course, trees and plants are very different to human or even mammalian cells and tissues, and the conditions they experience are also much warmer than the liquid nitrogen temperatures we are interested in here. However, they do act as a first indicator that living things can survive being frozen, and can survive being frozen in a large way. I think it's worth thinking about these tissues for a bit, and briefly cover some of the slow cooling and vitrification techniques which have been developed for these species for ultra-low temperature preservation. Plant preservation is usually focused on seeds or embryonic plants a few millimeters large. The same problem is often encountered as with human tissues: dehydration. If these embryonic plants have a high intracellular water concentration, they die. Some cells, after dehydration, can be cooled at a slow controlled rate and stored. However, vitrification is becoming much more common here, typically with high concentrations, up to 7 or 8 molar of glycerol and sugars, and rapid cooling. This eliminates ice damage, although has associated toxicity issues, a nice analogy to human and mammalian systems. What else can we learn from nature? I won't go into too much detail here, as this is covered much more rigorously in other talks at the conference, but we note that even when we move away from the more distant plant world to the animal kingdom, that here too we see freezing, or at least low temperature preservation of animals, all tissues intact. There are the famous tardigrades, which can survive freezing for decades without much apparent damage. There are frogs, such as Rana sylvatica, which pump their cells with sugars to survive being frozen solid. And there are then Arctic and Antarctic fish, which live quite happily below 0 degrees Celsius, using antifreeze proteins to protect against ice forming. All of these methods are under investigation as a key to organ and tissue preservation in humans, and they offer tantalizing proof that complex living systems can survive freezing in the right conditions. So, before moving on to human and mammalian species and current research, what other positives can we take from nature and past work? Well, the fundamental problems are well understood. Cell suspensions, in general, survive cryopreservation quite well. When systems become larger and the macrostructure damaged by ice, then the whole biological dies. We know that the whole biological macrostructure can be preserved, but at the expense of individual cell viability. However, focus on the individual cell viability and the macrostructure is lost. The solution must be avoiding ice, at least in its current form, in order for the tissue or organ to survive. However, we know that cell suspensions can be cryopreserved for most cell types, whereas larger tissues or organs cannot. Where does the boundary lie between survival and death? That will be the subject of the next section of my talk. Now, let's start the scaling up by looking at organoids and cell therapies. Spheroids and organoids are perhaps the smallest large tissues. A quick note on terminology here. When I talk about spheroids, I'm referring to clusters of cells up to around a few hundred microns across and consisting of a few hundred different cells of the same cell type. Organoids are similar, sometimes up to a few millimeters, and often more complicated with a range of different cell types. However, in general, these terms are used interchangeably and freezing strategies are similar. Although of course with multiple cell types, this does add an additional complication when it comes to optimizing cooling rates. Let's start with why we'd want to cryopreserve these at all. What advantage would we get from cryopreservation in these systems? The answer to this question is twofold, and it really depends on how the spheroid or organoid is formed. The first source are naturally formed in the body, a good example being pancreatic islets. Islet transplantation is often carried out from a deceased donor to a recipient suffering from type 1 diabetes. Often more than one transplant is required, and so cryopreservation allows us to ensure that we use effectively every last bit of this precious and limited resource. The second answer, why we need to cryopreserve these, is related to manufacture of bioproducts. On-demand manufacture is often not possible on cost and logistical grounds, and so cryopreserving manufactured spheroids and organoids allows for their clinical delivery. The fundamental reason why we want to cryopreserve these systems is consistent with why we always want to cryopreserve, namely to save some biological life from where it happens to be to where it is needed. Now let's see how these respond to cryopreservation. This wouldn't be a talk about cryopreservation if I didn't at least mention the two-factor hypothesis, which is shown on the figure on this slide. Essentially, that is, cooling must be slow enough to allow cells to dehydrate, but fast enough so that the toxicity of the cryoprotectants and other intracellular solutes does not become excessively damaging. How does moving from a single cell suspension to clusters of cells change this? Well, in general, in systems where ice is present, Moving from a single cell to an organoid will impact the rate at which cells can dehydrate. As opposed to cells simply losing water to the extracellular space, in organoids, cells will often lose water not to the extracellular space directly, but to the cell beside them, and so on until the edge of the sphere is reached. The larger the cluster of cells is, the lower the surface area to volume ratio, and so the slower cooling must be to allow sufficient dehydration. I will of course caveat this all with saying that each cell type has a different tolerance to toxicity and dehydration, and what may work for one cell type may not work for another unedited. This makes organoids consisting of multiple cell types more tricky, as parameters which are close to optimal for all the essential cell types must be found. In practice, we can see the impact of spheroid size on the cryopreservation outcome. The figure here is some work on Hep G2 liver spheroids of different sizes. The black bars are small spheroids consisting of between 20 and 50 cells, and as can be seen, the optimal cooling rate has quite a flat peak, the optimal rate around 2 degrees a minute, and that's similar to the optimal cooling rate for cell suspensions of this cell type. In grey, these are larger spheroids, around 100 to 150 cells. Here, the cooling rate becomes much more important, and slower cooling, around 0.3 degrees a minute, is optimal. Slower cooling will also impact the ice structure, and that's something I'll discuss in more detail in a few slides' time. Of course, it's not just when scaling up during slow cooling cryopreservation that size is a problem. The exact same issue is seen during vitrification. When it comes to vitrification, we again have a two-factor cause of cell death, although not exactly the same one as in slow cooling. The choice here is between cryoprotecting concentration, and so cryoprotecting toxicity, and cooling rate. To vitrify a system with, say, a typical 10% DMSO concentration, the cooling rate must be very rapid, hundreds of degrees a minute in normal circumstances. However, these rates are very difficult to achieve for anything greater than the smallest sample volume. With anything larger, you can get away with lower cooling rates, but only by increasing the concentration of cryoprotectants. These cryoprotectants are more toxic at higher concentrations, and an additional complicating factor comes from diffusion of cryoprotectant into tissue at high concentrations. Before we move on to whole organs and tissues, I will perhaps mention an important point of phrasing. Usually, the two differing cryopreservation techniques are called vitrification and slow cooling. I personally don't like the term slow cooling, as vitrification techniques can use slow cooling and ice can form while cooling quickly. However, I'll use them in this presentation due to their familiarity. Now, where are we with organs? Well, one of the most complicated systems on this scale, which is not normally considered an organoid, but for cryopreservation purposes match the definition I gave at the start of this talk well, are embryos. And the advantage of embryos is that they are perhaps one of the most studied cryopreservation systems out there. This is a really good example that shows the power of vitrification, and see the figure from Cook on this slide. Almost all embryos that are cryopreserved now in reproductive medicine are done so through vitrification techniques. The limitation is scaling up to organ-sized structures. So, onwards to organs and slow freezing. What is the state of the art here? The first thing I'd like to say in this section is that organ cryopreservation is not just some future aim. It can be and is done in selective systems at the moment. Two of the most notable organs which can currently be frozen, either in slices or as a whole, and survive happily, or at least as happily as you'd expect from something removed from the body, filled with non-physiological chemicals and put into a state of suspended animation, are ovaries and the thymus. And I'll look more into the thymus over the coming slides. The first thing to consider is what is special about these organs compared to others. What do they have in common? Well, the answer to this is in their physiology and makeup. Unlike other organs and tissues, such as the heart, these organs can survive quite happily if bits of it are cut off. The macrostructure is not critically important. Let's look at one of these organs, the thymus, in some more detail. In cryobiology, we often say that ice damage is the reason why we can't cryopreserve most whole organs using ice-forming techniques. But how do we actually know this? One of the best bits of evidence is that organs that can survive being sliced up are the ones that can survive ice damage and cooling. This also offers a possibility that if ice can be modulated, we may have a solution. Let's start by looking at one of the successes of large volume cryopreservation, the thymus. I'll start by giving an overview of what the thymus is and why we want to cryopreserve it. The thymus is an organ of the immune system where T cells mature. It sits just above the heart and in babies is not too much smaller than the heart itself. As you age, the thymus begins to be replaced with fibrous tissue, and by reaching adulthood, its size and activity is much reduced. Several medical conditions can affect the thymus, one notable one being DiGeorge syndrome. In such a case, which is apparent soon after birth, the thymus does not work properly or at all. Luckily, thymus transplants can solve these problems. Large parts of the thymus are often removed in patients who undergo neonatal heart surgery, and one thymus can be transplanted into more than one patient. Cryopreservation would allow us to preserve every thymus which becomes available, increasing supply and also allowing more rapid surgery for patients presenting with DiGeorge syndrome. And this is a project I've been working on with some collaborators at Great Ormond Street and UCL Institute for Child Health in London. As it turns out, cryopreservation of this system is quite straightforward, and the slicing of the thymus into strips, as is required for transplantation, is also helpful for freezing, these strips being about 0.5 mm thick. It allows cryopreservation with only minimal autolysis, and we're actually working on this right now and the results are promising. We have found that cooling at around 1 degree a minute is optimal for this system, and ice nucleation is not required. On thaw, we have some patch errors of autolysis, but a good outcome has generally been observed. This slide gives an overview of how the sliced organ system works in practice and some results. The thymus is removed from the donor, sliced into segments, cryopreserved, and then transplanted after thaw. The transplant actually happens into the thigh, as the location of the thymus in the body isn't too important for it to function, and thigh surgery is much safer than chest surgery. This gives an example of how, for specific organs, by using the advantages that the organ offers, in this case its ability to survive being sliced, they can be cryopreserved. However, what about the case of organs, say the kidneys, liver and heart, where slicing up is not really an option? What can we do there, and where do the limits lie? Well, to answer that, we must first acknowledge that for most cryopreservation of whole organs and tissues, where both cells and structure survive, it is not possible, at least not outside very experimental settings at the moment. But if we know ice is the problem, will it ever be possible to preserve an organ in the presence of ice? I am going to propose to you that the answer to that question is yes, or yes for some organs at least, and the key might be in the ice structure itself. I have here 3 cryomicroscopy images taken at -100 degrees in a solution of CS10, essentially 10% DMSO and some cryopreservation friendly solutes. But each image was taken following different rates of cooling. The image on the left, image A, is following cooling at 10 degrees a minute. The centre image, B, is following cooling at 1 degree a minute, and on the right hand side, image C is cooling at only 0.1 degrees a minute. What we see is at very slow rates of cooling, the ice structure is completely different. Much fewer, but much larger ice crystals. This is what you'd expect physically. Large ice crystals are more thermodynamically favourable compared to small ones, so given enough time, these large crystals will form. In fact, if you hold a sample at a high sub-zero temperature from some time, this structure always appears. While cooling at 0.1 degrees a minute is very slow, we've carried out work on a range of mammalian cell types and found that the reduction in viability is very minor even at these slow cooling rates. Special systems, such as sperm and red blood cells, of course, need much faster cooling rates, but we've tested T cells, liver cells and CHO cells and found that the minimal cooling rates required are very low indeed. Now, you could imagine that the ice structure in image C would be better for large tissues and organs. Fewer ice crystals and fewer boundaries between ice crystals would mean less damage. Well, what happens to an organ structure when you cool at these very slow rates? The results of an experiment in embryonic organs where we used a very slow cooling rate, in this case 0.2 degrees a minute from -5, are shown on this slide. With the collaborators at UCL Institute for Child Health, we cooled mice embryonic organs, shown in the slide is kidney on the left, the heart in the centre, and the liver on the right. These were cooled very slowly, and as can be seen from the images, on thaw most of the structure is intact. In fact, the histology shows good intact macrostructure throughout each of the tissues, which is very encouraging. Interestingly, some other work published by Vitrification et al. Managed to freeze whole uteruses using 0.2 degrees a minute in 10% DMSO. In these experiments here, we used 12.5% DMSO. There is limited data out there about very slow rates of cooling, but the data which does exist is encouraging. Now, while these results are interesting, I must also highlight the limitations of drawing too much from these results. We were able to grow living cells from these organs, however, we did not try transplanting or growing them for any length of time, so it would be premature to say they've been successfully cryopreserved in the true sense. These are also very tiny organs, the liver, the largest, only being about 5 millimeters in its largest dimension. However, what it does show is that with slow cooling, large structural damage can be avoided and, at least immediately on thaw, this can be avoided in complex systems. Before I move on to vitrification in larger systems, I wanted just to quickly mention an area that is sometimes overlooked in tissue and organ cryopreservation, and a system that could act as a halfway house to give incidental results along the way. This area is biopsies. Biopsies are small bits of tissue, typically no larger than a few millimeters cubed, which can be taken for medical tests, but are then often stored in large biobanks. These are incredibly useful, especially in population-wide studies. In such studies, tissues can be taken from a healthy individual. This healthy individual may go on later to develop a disease or condition, and the biopsy can then be accessed to determine if there was any indication that the patient might develop that disease long before a diagnosis was made. Such studies can help develop early diagnostic tests, which help increase the treatability and reduce the impact of said condition. At the moment, biopsies are typically cryopreserved using very basic plunge cooling techniques, sometimes in the absence of even cryoprotectants. This allows cheap and easy preservation, but often only some DNA and RNA can be extracted. New methods which we cryobiologists develop could be used for preserving cellular material well enough to outgrow cells post-thaw, as well as structural and cell-to-cell connectivity maintenance, which would allow an immense increase in the amount of data and so medical advances which could be made from biopsies and biobanks. And of course, the larger the tissue that we can successfully cryopreserve, the greater the information on offer. Now, these advances could be through either better slow cooling or through better vitrification techniques. So, vitrification, ice-free methods, have their own problems when scaling up. For most vitrification, very small volumes are used, and so rapid cooling and warming is possible, while using only a small amount of DMSO or other cryoprotectant. However, to scale to larger volumes, and just due to the physics of the system, the cooling and warming rates, at least conductively, are going to be slower. To prevent ice formation at these slower rates, higher concentrations of cryoprotectant will be required. The higher these concentrations, the more toxic they are, and so the less effective the cryopreservation becomes. There are two ways around this: to try and reduce the toxicity of the cryoprotectant, or to try and increase the cooling and warming rates. We've heard a lot about both of these areas in this symposia, so I'll not go into details here. However, specifically with respect to organoids, we've seen success in the past few years. Immature neural organoids can be quite readily vitrified using very rapid cooling and warming techniques, although results are still elusive when moving to more mature ones where cryopreservation would be most impactful. One area where we've seen good progress recently is larger spheroid preservation. The figure on this slide is from work done by Eva Pushman, who I worked with at UCL a few years ago. While I was working with slow freezing, she was trying vitrification in the same cell system. She found that by using liquidus tracking, that is only adding higher concentrations of CPA at a less toxic, lower temperature, she was able to effectively vitrify these samples with good viability post-cryopreservation. Interestingly and encouragingly, she found that vitrifying larger spheroids using this technique was at least as effective as cryopreserving the smaller spheroids by using an effective mixing process. It is likely that some mix of the above techniques will be effective for larger volumes. Larger volumes require greater perfusion times and so have greater toxicity from the cryoprotectant. They also must be cooled and warmed more slowly, and so the toxicity and risk of damaging ice becomes greater. On the other hand, new warming techniques will help increase from the inside out the warming rates, and new methods such as better cryoprotectant and cryoprotectant mixes and perfusion techniques will reduce incubation times and time-dependent toxicity. Now, time to wrap up. So, we have seen that scaling up from cell suspensions or smaller systems to complicated tissues and organs is at the forefront of cryobiological success. There is no easy answer in either ice-present or ice-free cryopreservation. One of the most advanced areas currently is cell spheroid and immature organoids. These vitrify quite well and are used extensively in some labs today. Slow freezing is also effective, and we have seen for some examples, such as liver spheroids, that just lowering the rate of cooling can result in a marked improvement on cell survival. As we get to larger volumes, the negative impact of cryopreservation becomes more pronounced. Most of these systems cryopreserve poorly, and it's only the ones in which macrostructure is less important, such as ovaries and the thymus, where cryopreservation success can be assured. It seems a fairly safe bet that the ice structure itself is the problem, and this can be moderated by using very slow rates of cooling to change the ice structure, although ultimately in the future perhaps new methods in vitrification allowing rapid rates of warming and cooling and less toxic methods of DMSO will be effective. So, thank you all very much for listening, and before opening up to the question session, I'd just like to thank some of my collaborators who have been working on some large volume scale-up projects with me. I'd like to thank the team at UCL in Great Ormond Street, Tessa Crompton, Sue Ross and Meera Chowda for the work on thymus preservation and for the great images and data we got with the embryonic mice organ work which we did together. Graham Davies, who's been working on the thymus transplantation project, also at Great Ormond Street, and the Cytiva team who have been working on larger volume cryopreservation at our Cambridge site for a number of years. I hope you all enjoyed this talk, and I'll now open for any questions.